Welcome to Biochemistry at the School of Life Sciences, Keele University. I'm David Watson, Programme Director for Biochemistry, and this short presentation will cover some of the key features of our programme. This presentation will focus on why you should want to study biochemistry at Keele, and will include details on the course structure, indicative content, the teaching facilities including assessment types, and finally the types of career routes a degree in biochemistry may lead to, and some typical destinations of our former graduates. The biochemistry course is available as either a standard 3-year BSc degree accredited by the Royal Society of Biology, or, new for 2020, a 4-year integrated MSci, a master's level degree combining the standard 3-year BSc course with a 4th research-focused year of study, subject to validation. Applicants interested in the 4-year MSci route should contact University Admissions or the School of Life Sciences, but also note that you would have the option of transferring to this route during your course up to the end of the 2nd year of study, subject to satisfactory performance. You should also view other screencasts available from the school web pages covering the types of placements available to students across all of our life sciences programmes, including options for studying a semester abroad, or for turning your BSc course into a 4-year degree or MSci course into a 5-year degree with a year-long placement or year of international study abroad. The focus of our programme is very much on mammalian biochemistry and in particular the application of biochemistry to understanding human health and disease, aligning very much with the department's research strengths. Our course is structured over 5 core themes covering all topics recommended by the Biochemical Society and giving you a broad coverage of life in molecular detail. The first of these themes focuses on macromolecular structures and importantly the relationship between structure and function. This includes the formation of complex 3-dimensional structures of proteins and their interactions with other macromolecules, including other proteins or DNA, as well as their interactions with small ligands such as drug molecules. This theme also explores the complex structure of other macromolecules, including carbohydrates, and the field of glycobiology in understanding cellular processes and disease treatment. The second theme covers cell metabolism, from an understanding of the major metabolic processes for energy generation in cells through to the regulation of metabolic homeostasis and the conditions that may arise from a dysregulation of these processes, such as insulin insufficiency and the development of diabetes. Thirdly, the course will also develop an in-depth understanding of molecular biology and genetics. Building from our understanding of the molecular processes of DNA replication, transcription and translation can be exploited in application to molecular cloning and gene therapy in the treatment of disease. The fourth theme focuses on the structural organisation of the cell, including mechanisms of cell transport and subcellular organelle structure-function relationships, as well as exploring the processes of cell-cell communication and cell signalling, studying how hormones and other messengers can signal changes in cellular activity or behaviour. For example, the intracellular events that occur from a messenger binding to a cell surface receptor, leading to changes in gene expression or other cellular responses. Finally, as biochemistry really is the chemistry of life, the fifth theme on the applications of chemistry really underpins much of the other material, whereby we explore biological processes in molecular detail, applying concepts from the chemical sciences with very much a biological emphasis. For example, in developing techniques to determine the 3-dimensional structures of proteins by X-ray crystallography, and our knowledge of these structures informs drug development, a process of rational drug design, exploiting important structure-function relationships. Our programme is structured as a spiral curriculum whereby material related to each of those core themes is revisited at each level of study, progressively increasing in detail. The first year of the course is structured so that the modules will give you a good grounding of core knowledge in each of those theme areas. A list of these modules can be found on the course webpage. Alongside the core academic material, you will also develop a range of practical laboratory techniques and skills in data analysis from an integrated laboratory programme across many of your modules. Importantly, this allows the practical teaching to be very much aligned with what you'll be learning in associated lectures. For example, a lecture series on enzyme kinetics and the mechanisms of enzyme catalysis runs alongside a series of laboratory sessions and data analysis workshops where, working with an enzyme, you will explore the effects of manipulating various conditions on rates of reaction. The second year of the course will then revisit the core concepts from the first year, but in a more applied context. For example, knowledge of how proteins fold into complex 3D structures is applied to the structural organization and function of proteins involved in mediating the immune response, such as antibodies. This also draws on an understanding of genetics and molecular biology in understanding the great diversity of antibodies we produce to defend against bacteria, viruses, and other pathogens. This year of study will also further develop your laboratory skills, particularly with some core molecular techniques, as well as developing skills in experimental design, which also runs as an integrated laboratory program aligned with taught module content. This year of study will also introduce you further to the growing field of bioinformatics and the application of computational tools to the analysis of protein structures and in drug design. The third year of the course places a greater emphasis on the application of biochemistry in addressing today's research questions. Building on material from each of the 5 core themes, you will select from a range of optional modules alongside some core modules, exploring topics ranging from biotechnology and sustainability to the application of complex carbohydrates in the treatment of disease in medical glycobiology. Optional modules may include exploring the molecular changes associated with the development of cancer and how this may lead to novel therapies, or studying further the relationship between the macromolecular structure and function of proteins. Topics all align to areas of ongoing cutting-edge research activity in the School of Life Sciences. All students in the final year, if not on placement, and remember to see other screencasts for more on this, will undertake an independent research project under the supervision of a member of staff. The types of project available may range from lab-based research, either in one of the department's research labs or dedicated final year project multi-user facility, to literature-based systematic reviews or computational projects in the field of bioinformatics and big data. In addition to your project report, the culmination of the research project is the Undergraduate Student Research Conference, where students from across all undergraduate disciplines in the School of Life Sciences attend a full-day programme of student talks, poster presentations, and a careers-focused networking event attended by a range of potential employers. This is very much in the style of a professional research conference, which many of you may attend at national or international level in your future careers, or as part of further postgraduate study. For students taking the 4-year MSci course, the 4th year of the course is a research-focused year of study. Modules taken across this year will further develop your understanding of cutting-edge research methodologies and techniques and their applications in modern biochemistry through a combination of research-level seminars and small group tutorials. As part of this and other assessments in this year of study, you will also expand your skills from previous years in the evaluation and critical analysis of scientific literature and the assessment of research impact. All MSci students will also undertake an extended research project, further developing skills in experimental design, data collection and analysis, and the production of publication standard research outputs. The way you will be taught across the programme may vary between modules, but most modules will typically have between 2 and 4 hours of lectures per week. As our laboratory teaching is integrated across some modules, you will also typically have between 3 and 6 hours per week of laboratory classes or supporting tutorials and skills workshops, covering materials such as data analysis, report writing, or model building, directly supporting the laboratory programme. Some of our modules also incorporate small group case-based learning in groups of around 12. In the first year, these sessions are based on clinical scenarios where the focus is on the underlying biochemistry. These small group sessions gives you the opportunity to discuss and share your knowledge as a group, identify areas where further research is needed, on which you then allocate tasks and report back to the group in later sessions. Many of the cases used also directly support material being delivered in some of your taught modules. For example, a case study on cystic fibrosis leading to a more detailed understanding of the mechanisms of chloride transport and the genetics of hereditary disorders. You can see from this then that the structure of the programme means that you are taught in a variety of different ways, something which is also reflected in our assessment strategy. Modules will be assessed in a number of possible formats. Some will include an end-of-module examination, taking place either in January or May, depending on which semester the module runs. And most modules will also include elements of coursework. This can range from laboratory reports to essays, presentations, or developing a web page, and several modules may also include elements of group work, such as a group presentation or the collective contribution to an online activity. It is not surprising then that given the structure of our programme and the mixed and varied approach we take to teaching and assessment, that the biochemistry course at Keele University currently ranks in the top 10 nationally for overall student satisfaction in our subject category. We have a number of modern and well-equipped teaching laboratories in the School of Life Sciences, accommodating between 40 and 120 students, including the recently opened David Attenborough Laboratories, opened in person by Sir David in 2019. The size and capacity of these means that in most laboratory classes students will work in pairs or as a group of 3, working together to complete the session aims or dividing tasks between the group. Our teaching laboratories are also well supported by a team of dedicated technicians, subject-level senior demonstrators and postgraduate research student demonstrators, helping to support students alongside a lead member of staff. A degree in biochemistry gives you a wide range of future career options, which may include a career in the pharmaceutical or biotechnology industry, scientific sales and marketing, or in the development of laboratory reagents and equipment. Many of our graduates will also go on to further study at postgraduate level, including a master's course or PhD, or with further study in medicine or teacher training. Additionally, the analytical and generic transferable skills gained from a biochemistry degree are attractive skills and qualities for any number of career options, both within and external to the wider biosciences. The university has an excellent career centre, offering tailored support for students with writing CVs, application forms, interview technique, and general advice on possible graduate opportunities. To find out more about the structure of the biochemistry course at Keele, including indicative modules, visit the course web pages. Here you will also be able to enter a virtual chat with a current student to find out more about student life in the school. Alternatively, visit the Offer Older Day web pages, where you can also submit additional questions. Finally, I look forward to welcoming you to your studies in biochemistry at Keele.